السلام عليكم. ازيكم يا شباب؟ كل سنة وحضراتكم طيبين. إن شاء الله هنتكلم اليوم على الماسكولار تيشو، ودوت من أحد البيزك تيشوز الموجودة عندنا في our body. التيشوز دي include الإبيثيليوم أو epithelial tissue, connective tissue, muscular tissue and nervous tissue. السيلز الموجودة في الماسكولار تيشو دي تبقى optimized for contraction، وبالتالي بتساعد الموفمنت of particles or food inside our gastrointestinal tract أو GI tract. برضو بتساعد الموفمنت of blood inside our blood vessels. برضو بتساعد الموفمنت of our body as a whole. The muscular tissue include two types: non-striated or smooth muscle. The other type is a striated muscle, and this includes skeletal muscle. This type of muscle is under your control, while the other type, or cardiac muscle, is not under your control. The three types of muscle fiber can be distinguished according to morphological and functional characteristics. On the left we have the skeletal muscle fiber. الفايبر بتبقى متجمعة في هيئة bundle of elongated striated fiber. These fibers are multinucleated. Their nuclei are peripheral in position. The contraction of the skeletal muscle is quick, forceful, and under your control. On the right we have the cardiac muscle fiber. They also form bundles. Cardiac muscle fiber often branch, processes from one cell, join processes from the other cell at a junctional complex called intercalated disc. Unlike skeletal muscle, the nuclei of this muscle fiber are central in position. Their contraction is vigorous, rhythmic, and involuntary. On the middle we have a collection of fusiform non-striated cells of smooth muscle fiber, and their contraction is slow and involuntary. خلينا نبدأ بالskeletal muscle. Skeletal muscle as a whole is surrounded by dense irregular connective tissue called epimysium. The epimysium carries large blood vessels, lymphatics, and nerves of the muscle. The muscle is composed of several bundles of muscle fibers. Each bundle is surrounded by a thin layer of connective tissue called perimysium. Each fiber in the bundle is surrounded by a delicate layer of reticular fiber with scattered fibroblast called endomysium. The endomysium surrounds the external laminae of the muscle fiber. Within the endomysium, we can see the blood vessels and nerves that supply the muscle fiber. In longitudinally sectioned muscle, the muscle fibers are parallel to each other. Their cytoplasm, also called sarcoplasm, is acidophilic and shows cross striation. Their nuclei are elongated and peripheral in position. In transversely cut muscle, the muscle fibers are polygonal and of varying size. Their nuclei are peripheral in position and they appear rounded. Let's have a closer look to the banding pattern of the skeletal muscle. As we see here, there is an alternating dark and light band. The dark band is anisotropic and is called A band, while the light band is isotropic and called I band. This banding pattern is actually on the myofibrils, and because the myofibrils are arranged with these bands are aligned, This banding pattern is reflected in the muscle fiber as a whole. Under the electron microscope, the sarcoplasm of the muscle fiber is occupied by myofilament containing myofibrils. These black lines delineate the boundaries of these myofibrils. Myofibrils show dark band or A band and light band or I band. The A and I banding pattern is due to the arrangement of the myofilament within the myofibrils. At the center of the dark band, there is a pale area called H zone, and the light band is bisected by Z line. Let's take this black line out of our way. In the intermyofibrillar region, as the I band, you will see the mitochondria and refer to it by this letter M. The lower figure shows parts of three myofibrils. In the intermyofibrillar region, we see the glycogen granules, these black dots, and together with lipid droplets, they provide muscle with energy. Additionally, in the intermyofibrillar area, there are tubules belong to the sarcoplasmic reticulum tubule system, and we will talk about this part in minutes. The sarcoplasm also contains the oxygen-carrying pigment myoglobin, and this myoglobin contributes to the red coloration of the muscle fiber. In the next three slides, we will focus on the structure of the myofibrils. Here we have the skeletal muscle fiber, and this fiber is composed of several myofibrils. Let's focus on this myofibril. As we see here, it's composed of repeated functional subunit called sarcomere. Each sarcomere lies between two Z line or Z disc. Let's focus here on the sarcomere. The sarcomere contains two types of filaments. Thick filament, which is composed of myosin and present at the center of the sarcomere, and they contribute to the A band. The thin filament and formed of actin filament, and they form the I band and extends into the A band. This area, labeled by the orange asterisk, represents the area of overlap between the thick and thin filament. At the middle here, we have the H zone. In the H zone, there is a protein that cross-link myosin in transverse direction, and these proteins appear as line at the center of the H zone called M line. Z lines are formed of alpha-actinin filament and other material, and they gave rise to the attachment of thin filament in both sides. Here we have this green structure called titin filament, and this titin filament links the thick filament to the Z line. Titin filaments prevent overstretching of the sarcomere. Also, they maintain some sort of overlap between the myofilaments. In the next slide, we will talk about the molecular structure of the myofilaments. The thick filament is formed of myosin molecules. This large molecule is formed of two identical heavy chains and two pairs of light chains. The heavy chains are twisted around each other to form the tail, while the light chains form the globular head and extended at either end of the heavy chain. The mucin head contains actin binding site, ATP, and ATPase binding sites. Several hundred of these mucin molecules form a thick filament. The actin filament is a helical filament and formed of F-actin. Each F-actin is a polymer of G-actin monomer. In each G-actin, there is a mucin binding site. There are two proteins associated with the actin filament, tropomucin and troponin. The tropomucin is located in the groove between the actin filament strands. The troponin complex is composed of troponin T, which binds tropomucin, troponin C, which binds calcium ion, and troponin I that regulates the interaction between the actin and mucin. Unlike other cells in our body, the smooth endoplasmic reticulum of the skeletal muscle fiber forms an elaborate membrane system called sarcoplasmic reticulum. At the A band, the sarcoplasmic reticulum gives parallel tubules. These tubules form fenestrated cisternae at the H zone. On the other side of the parallel tubules, at the AI junction, these tubules form terminal cisternae. Invagination of the sarcolemma, referred to them by these arrows, gave rise to transverse or T tubules. These tubules run between two terminal cisternae. Numerous gap junctions are found between the T tubules and terminal cisternae to facilitate the propagation of action potential from the T tubule to the cisternae. This arrangement of a T tubule surrounded by two terminal cisternae referred to as a triad. Similar arrangement of the sarcoplasmic reticulum is seen at the Z line. As sarcoplasmic reticulum gives fenestrated cisternae at the level of the Z line and terminal cisternae at the A junction. Two terminal cisternae together with a T tubule gave rise to a triad. Skeletal muscle fibers receive motor supply by myelinated axons. After passing the perimysium, these axons gave rise to unmyelinated small branches but covered by Schwann cell. Near the muscle fiber, the axon terminal expands, and this region is covered by Schwann cells. The external lamina of these Schwann cells is continuous with the external lamina of the skeletal muscle fiber. The expanded axon terminal contains mitochondria and vesicles with acetylcholine neurotransmitter. Underneath the expanded axon terminal, there is a synaptic cleft. In the other side of this cleft, there is a postsynaptic membrane. This membrane undergoes junctional folds to increase the surface area of the membrane and hence the neurotransmitter receptors. When the nerve impulse reaches the axon terminal at the neuromuscular junctions, it triggers the release of acetylcholine. Acetylcholine passes through the synaptic cleft to bind its receptor in the postsynaptic membrane. This initiates an action potential that propagates along the sarcolemma and goes through the T tubule to trigger the release of calcium ion from the terminal cisternae. Released calcium ions in the sarcoblast bind troponin C with the troponin complex. This initiates conformational changes. Ultimately, tropomyosin moves away, exposing the actin active sites. Myosin heads bind actin active sites to form cross-bridging. Then, myosin heads pivot, moving the actin filament to the center of the sarcomere. Hydrolysis of the ATP molecule detaches myosin heads from the actin active sites. Myosin heads return to their original position. This cycle is repeated several times as long as calcium ions bind with troponin C and exposing the actin active sites. Now, what would happen if you don't have ATP? If you don't have ATP, as in after this, the myosin head continue attaching to the actin active sites. This state is called rigor mortis. After cessation of the action potential, calcium ions are pumped back to the sarcoplasmic reticulum and tropomyosin moves back, covering the actin active site. Actin filament return passively to their relaxed state. Let's focus on changes occurring in sarcomere during muscle contraction. The upper part of the figure demonstrates sarcomere in a relaxed muscle, while the lower part of the figure is showing a sarcomere during contraction. Do you remember these? These are the titin filament. During relaxation, the titin filament maintain areas of overlaps between the thin and thick filament. This area is equivalent to the areas labeled by the yellow asterisk on the right-hand figure. Also, during relaxation, the H zone is prominent. On the other hand, upon muscle contraction, actin filaments move towards the center of the sarcomere. This brings the Z lines closer together and compress the titin filament. On the right-hand figure, we will see the sarcomere becomes shorter than its state during the relaxation. Each zone is gone. The I band becomes shorter than before. However, the A band is constant. The A band does not change during contraction. Each muscle in our body has different flavor of muscle fiber types. We have 3 types of muscle fiber: red or slow, wide or fast, and intermediate muscle fiber. The content of this muscle fiber in a given muscle depends on the activity performed by this muscle. For example, rapidly contracting muscles like tibialis anterior, they have a lot more wide or fast muscle fibers than red or slow fibers. On the contrary, the soleus muscle responds by slow but prolonged contraction, and they have a lot more slow muscle fibers than wide or fast fibers. Here, I'm going to highlight the differences between these types of muscle fibers. Let's start with the red muscle fibers, also called slow oxidative muscle fibers. These fibers are the smallest in diameter. They perform aerobic respiration. This means that they need oxygen. Oxygen is supplied by the extensive capillary networks these muscles have. Oxygen is stored in myoglobin, which contributes to the bright red coloration of these muscles. They are highly populated with mitochondria for high rate of oxidative phosphorylation. They are slowly contracting and resist fatigue. On the other hand, the white, fast or glycolytic muscle, they are the largest in diameter. They have a large sarcoplasmic reticulum for rapid release of calcium and and contraction. They have a lot glycolytic enzyme for rapid release of energy. They have much less blood vessels and mitochondria because the oxidative phosphorylation is of secondary priorities. They are deficient in the myoglobin, therefore they are red. This muscle fiber type fatigues easily. The third type, which is intermediate muscle fiber, have the characteristic in between the other two types. Second type of muscle we are going to talk about is the cardiac muscle. Similar to the skeletal muscle, cardiac muscle is composed of striated cylindrical fibers. Unlike a skeletal muscle, cardiac muscle fibers are branch-shaped fibers. Branching from one fiber join branches from others. They have pale staining centrally located nuclei. A unique feature of the cardiac muscle is the presence of transverse lines that cross the muscle fiber at regular intervals. These lines are called intercalated discs and refer to them here by the letter I. We are going to talk about the intercalated disc in minutes. Ultrastructurally, the sarcoplasmic cardiac muscle fibers show myofibrils with the same banding pattern of the skeletal muscle fibers. The intermyofibril region shows abundance of mitochondria, and this reflects the metabolic demand of these cells. The transverse tubule or T-tubule system here is less organized, and they usually associated with the unexpanded terminal cisternal sarcoplasmic reticulum. This is referred to as dyads instead of triads of skeletal muscle fibers, and these dyads are located at the level of the Z lines. Contraction of the myocardium is initiated by the SA node. The action potential extends from the SA node through the atrial myocardium to the AV node. From the AV node, pass through the right and the left bundle branches. The distal process of the bundle branches are called Purkinje fibers. These fibers are located in the subendocardial region of the ventricular wall. Let's look to the left-hand figure. The area labeled by these two arrows is the subendocardial region. Fibers in this region are larger than the contractile fiber of the myocardium. These fibers are Purkinje fibers. Let's go to the right-hand figure. Fibers labeled by these arrows are larger than the contractile fiber in the rest of the myocardium. They appear pale, and they contain one or two nuclei, and the cytoplasm is filled with glycogen. Under electron microscope, the sarcoplasm is Purkinje fibers rich in mitochondria, and while myofibrils are sparse and push to the periphery of the sarcoplasm. At point where branches of the cardiac muscle fibers meet, the cell membranes become associated with each other, forming junctional complexes. These are the intercalated discs. They appear as transverse lines. These lines cross the muscle fibers at regular intervals. If we look to the lower figure, the intercalated disc is arranged in a step-like fashion. It has transverse portions and lateral portions. The transverse portion run across the myofiber, while the lateral portion runs parallel to the myofibers. On the transverse portions, we have here zonular adherents, and these zonular adherents give attachment for the thin filament of the terminal sarcomere. Also in the transverse portion, we have macula adherents or desmosomes, and these desmosomes hold the cardiac myocyte together and prevent them from separation during vigorous contraction. Gap junction is found here in the lateral portion of the intercalated disc. The third type of muscle in our body is the smooth muscle fibers. They appear as a collection of spindle-shaped cells. They are broad in the middle while tapering towards their ends. They are of different sizes, with the largest are found in the wall of the gravid uterus. Their nuclei are spiral in shape and are present in the center of the wide portions of the fiber. To achieve the tightest packing, broad portions of the fibers are usually opposite to the tapered region of the adjacent fibers. Adjacent fibers become intimately attached at certain point by nexuses. Organelles of the smooth muscle fibers are mainly located in small areas on both sides of their nuclei. They include mitochondria, Golgi, rough endoplasmic reticulum, and free ribosomes. The rest of the sarcoplasm is filled with myofilaments. There are dense particles into which some subfilaments are inserted. The sarcolemma is invaginated to form little caves or cavuli. These cavuli get contact with the widely distributed smooth endoplasmic cisternae localized beneath the membrane. There are three types of filament in smooth muscle fibers. The first is thick and is formed of myosin, and the second is thin and is formed of actin. Let's look to the upper right hand figure. Thin filament is attached to dense bodies. These dense bodies are formed primarily of alpha actinin. They are scattered in sarcoplasm or attached to the sarcolemma. The sarcolemmal dense bodies also are the site of attachment of intermediate filament formed of desmine and vimentin, and the site of adhesive junction with other cells. In the reaction smooth muscle cell, contractile units extend from side to side. Upon contraction, thin filament anchored to dense bodies in the sarcoplasm slide over the stationary thick filaments. This twist and squeeze the smooth muscle fiber in a corkscrew action. All muscle fibers are derived from myoblasts. In skeletal muscle fiber, myoblasts fuse end-to-end to form multinucleated muscle fiber. Skeletal and cardiac fibers cannot divide because they lack centrosomes essential for cell division. However, smooth muscle fibers can divide. Skeletal muscle have a special type of undifferentiated cells called satellite cells. They are located between the fiber and their basement membrane. Under certain conditions, satellite cells divide and differentiate into skeletal muscle fibers. These fibers may join existing fibers to lengthen it or replace damaged ones. Satellite cells are absent in cardiac muscle. Therefore, when cardiac muscle is exposed to injurious factor like myocardial infarction, they heal by formation of connective tissue scar.